welcome folks. This is workshop 8, which is the final workshop and the last session before the closing remarks. So workshop 8 is going to be about principles of genetic circuit design, which is personally very close to me because this is what I did as a PhD student. So I'm very excited for this workshop. It's going to be given by Hatem, who's a research assistant at Zeval City of Science and Technology. So with that, Hatem, take it away.
Yes, thank you, Prashant, for this introduction. So today, my name is Hatem. First of all, my name is Hatem. I'm a research assistant at Zeval City of Science and Technology. I'm also a judge and a mentor of 2 IGM teams and a member of the IGM Engineering Committee. I also have recently received the Global Community by Fellowship from the Global Community by Summit. Today I will present to you a workshop on the principles of genetic circuit design. So let's start. So the outline for the workshop will covers the foundation and the application of genetic circuits, and also modular composition of genetic circuits from different classes of gene regulators. How can we compose genetic circuits from different types of regulators? Also, the genetic circuit design workflow, which I will cover from design specification till build optimization. And also I will finally cover the common failure modes encountered in genetic circuit design and engineering strategies to overcome these failure modes. So let's start. A brief introduction about genetic circuits. So genetic circuits are basically regulatory interactions that enable cells to make decisions and to integrate signals from its environment. They control gene expression in the levels of gene expression, the timing, conditions of gene expression. As I mentioned, cells process information from the environment. They integrate these information through genetic circuits. For example, we have in the cell different types of transcription factors. These transcription factors can sense different types of environmental signals. And based on this, the activated transcription factors could activate or repress the expressions of certain genes inside the cell. So these kind of interactions we call regulatory interactions, we call genetic circuits, because these transcription factors can also activate other transcription factors and initiate other kinds of gene expression. In this case, genetic circuits can control the sensing and response behavior of the cell. There are basically for the transcription factors, there are 2 types, like activators and repressors. Activator basically activates gene expression in response to specific signals, while repressors can repress gene expression in response to specific signals. And typical time scales for such regulatory interactions, as outlined in the table at the bottom of the slide, the binding reaction of a small molecule to transcription factors can be very fast on millisecond time scale. Also, the binding of the active transcription factors to DNA site to initiate gene expression on the second time scale. The transcription and translation can occur in minutes. However, the accumulation, which is the slowest part of the process, the accumulation of the protein product can take an hour in order for a protein to reach a steady state concentration. So this is an example of genetic circuit used by phage to decide whether to go for lysis or lysogeny. When the phage infects the cell, it can make 2 decisions whether it can proceed with lytic busway and make copies of it, and or it can integrate into the bacterial genome, and in this case it remains dormant until it make a lytic decision later on in its life cycle. So basically we have a genetic switch encoded in the feed genome that enables it to make a decision whether to lysis, whether to go for lysis or lysogeny. This decision circuit is encoded by transcriptional repressors, and they sense signals like the state of the growth, the state of the host, and based on this takes a decision whether to and also the temperature, whether to make lysis or lysogeny. This is an example of a natural genetic circuit that make decisions. So we have genetic circuits. In order to study these genetic circuits, to predict its behavior, to analyze its behavior, and to also to engineer its behavior, we need to model these genetic circuits. We cannot talk about interactions in a narrative way. Rather, we describe these interactions in mathematical equations, and use these mathematical equations to model, to simulate and predict the behavior of genetic circuits. So we start basically with genetic circuit topology, which is a regulatory structure of the regulatory interactions in the circuit, and we make a list of the biochemical reactions that happens in this genetic circuit, and based on and use these biochemical reactions to construct a mathematical model which can be reduced to different strategies and then simulated. The benefit of a reduced model is that we have fewer parameters and can be easier to analyze and can be easier to predict parameters from experimental data. However, in a detailed ODE model, by the way, is describes the rate of production of proteins, the rate of production of a chemical species, for example, a protein, in terms of its kinetic parameters, in terms of the concentration of reactants. Like, for example, the protein, the rate of production of protein would be controlled by the rate of transcription and the rate of translation, and also will decrease by the rate of protein degradation or mRNA degradation. So basically, ODE model captures these biochemical reactions in mathematical equations that can be modeled and simulated. So I hope that this is clear so far. If you have any question, you can go ahead and ask. Hello? Yes. Anyone has any question? Okay, let me proceed. So for the as I told you, we have the mathematical model of the, once we mathematically model this biochemical equation using ODE model, we use different type of strategies to do this model with fewer, in order to reach a simplified model with fewer parameters that is amenable to analysis. A common form of the reduced model is a Hill function. A Hill function basically describes the rate of production of protein in terms of the concentration of the activated transcription factor, whether it is an activator or a repressor. So we have this Hill function. This is a common form of the reduced model that can simplify the modeling and the analysis of genetic circuit. And there is also a logical approximation for this reduced model. This logical approximation basically describes the activation of the gene expression in terms of on and off states. However, in the health function, you can have graded gene expression. You could have continuous levels of gene expression. However, in logic approximation, you have on or off state. So the modularity of the regulatory interactions of genetic circuits enable forward engineered genetic circuits. You have in genetic circuits promoters. You have regulators like ribosome binding sites. You have coding sequences. You have terminators. You can just rewire these components in order to build novel behavior. As I mentioned before, that the cells integrate signals, processes signals, and make customized response. We could program this sense and response behavior by simply rewiring these regulatory paths together. Forward engineered genetic circuits, examples of forward engineered circuits, or the first examples of genetic circuit engineering are toggle switches, as well as reversator. Toggle switch that can just program two different states of genetic circuits, whether on a state or on or off, based on input signals. And also the reversator, the oscillator, genetic oscillator that can simply generate oscillation in gene expression. This oscillation can synchronize, for example, downstream biochemical reactions. Such control over gene expression allows to program behaviors that are of therapeutic purpose, of therapeutic significance, or for biotechnological significance. These are our examples of the potential of synthetic genetic circuits, of engineering genetic circuits. We have, for example, examples for engineering bacteriophage to treat infectious diseases, also for antibiotic resistance, and also for engineering bacteria to invade, to detect cancer, invade cancer, and attack cancer. And also we have examples of rewiring metabolic cellular and the metabolic pathways in the host in order to restore homeostasis, for example, restoring glucose homeostasis and things like that. So,
excuse me,
yes.
Can you go back to the previous slide?
Yes.
Oh. The thing which is explained at the right bottom, can you explain it a bit again?
Come again?
The network rewiring thing.
Yes. Let's talk about, for example, rewiring. You mean the rewiring in the host, right?
Yes.
Okay. In this case, you could have, for example, a pathological malfunctioning network inside the host. For example, you don't have production of a specific important molecule. And in this case, you could interface a synthetic genetic circuit that detect these pathological conditions on the host. And based on these pathological conditions, you can trigger the expression of this therapeutic molecule. So you simply couple the expression of a therapeutic molecule to the conditions of the disease. Is that clear?
Yes, thanks.
Okay. So let's talk about how to design genetic circuits. Let's start with the automation. With the automation of genetic circuits. Actually, this is not full mature yet, but let me give you an idea about how the automation of genetic circuit would work, and also the importance of genetic circuit design automation. First of all, the generic circuit, as is generic circuits, when the generic circuits are engineered to make complex functions, the complexity of the interaction increases, and in this case, you need some sort of abstraction in order to rather than thinking about the sequence, you can think about how to design, how to, for example, couple genetic modules or couple genetic devices to build more complex functions. If you think about the sequence, you can only think about designing simple functions. I would think about, for example, let's say differentiate pattern forming genetic circuits. In this case, you need to couple genetic modules that, for example, triggers a differentiation of cells that, for example, interpret morphology ingredients. These genetic modules, to couple these genetic modules, you need tools to couple these genetic modules and predict their behavior and to automate this process. So we have enormous potential of synthetic genetic circuits, and to harness this potential, we need more abstraction in our genetic design workflow and more automation. Computer-aided design tools help in this design. Up till now, there is a computer-aided design tool that is fully automated called Cello. This simply takes high-level language, high-level design specification described in high-level language, and combines this high-level language into DNA sequence. This is basically how a genetic computer-aided design tool for genetic design automation would simply work. You simply input high-level specification of the genetic circuit, and you have as output a DNA sequence that encodes the function encoded in the high-level language. So you start with the specification and you end with a DNA sequence. This is a fully automated process. But up till now, there are only a few tools that could do this, like Cello. But the other tools available are not yet at this scale. But there are different tools, for example, for the specification, there are for the modeling, for the simulation, like Toolchain, but you don't have a fully automated CAD, except for Cello, for example. So this is the Cello, the workflow of the Cello. Cello, by the way, is specialized in designing genetic logic circuits. You simply specify this behavior of the circuit in a language, hardware programming language called Verilog, and Cello then uses a library of genetic logic gates. First, it compiles this high-level specification into a truth table, into Boolean logic expressions, and use these Boolean logic expressions along with a library of genetic logic gates to build the final genetic digital circuit. And this using already a genetic logic gate with a non-sequence, because this is already fully characterized. So once you just connect the logic gates, you arrive at the final sequence. You can do then some sort of optimization of the sequence, but the full automation of this tool allows to design more complex genetic circuits that can make more complex digital operation inside the cell. But as I mentioned, this tool is used for designing of computational operations, digital computational operations. But we don't have tools, for example, for designing analog operations, which are analog circuits, by the way, are useful for making computation at lower resources. However, digital circuits require more resources, which can be an overload on the host. So this is examples of digital logic circuits. On the left side, digital logic circuit designed by Cello, and this match the simulation of these, the behavior of these circuits, match their experimental data. So this was a successful example of a genetic design automation tool. So how to design a function inside the cell. Before I speak about genetic design workflow, genetic circuit design workflow, I need to explain to you generally how to design a function inside the cell. Basically, you start with a high-level description of the function that you want to encode. For example, you want to program a bacteria to seek and destroy cancer cells. So this is a high-level specification. You need to make some sort of top-down decomposition of this high-level specification in order to encode this function in modules, rather than you have a fully complex function, and you need this fully complex function to be encoded by smaller functional modules. And these smaller functional modules will be encoded by genetic sequences. So in this case, you are breaking down your complex design process into simpler design cases. So you do some sort of top-down decomposition, and you constrain your functionality. For example, you want a bacteria that invade cancer cells. So you need motile bacteria. You need bacteria that can survive cancer conditions. So these sort of design constraints, you need to specify them as well in order to arrive at a functional system at the end. And once you make this specification and top-down decomposition, you can then start searching for genetic modules that encode your sub-functions. So, for example, in our case, for a bacteria invasion system, you have these functional modules. You need a bacteria that can sense, that can first fuse into the cancer cell wall, into the cancer cell the plasma membrane, and sense the vacuole condition, because it will be in endocytosis into vacuole, sense the vacuole condition, and based on this sensing can trigger the expression of vacuole lysis genes and also self-lysis genes, and so that it can degrade and it can release its payload to the cancer cytoplasm. And they can also do another function due to do cell spreading. So these you just breaking the complex function into simpler function modules, and you search for genetic sequences that encode these sub-functions. So let's talk about how to design a genetic circuit that, for example, in this case, could control the level of expression of these genetic modules for our engineered function, the timing and conditions. You can couple the expression of these cancer invading function modules to the cancer environment conditions, so that you can, for example, you can sense pH, you can sense hypoxia conditions. You can couple these conditions to your engineered function. So you need a genetic circuit to control the function. For example, you can engineer an AND gate that in the presence of low oxygen conditions and in the presence of low pH, you can just initiate the gene expression of your cancer invasion functionality. So simply, genetic circuit makes this a kind of control. So this is a basic workflow of how to design a genetic circuit. You start again with the specification. You specify the input-output behavior of the genetic circuit. And also, if you are thinking about dynamic circuits, you can specify the dynamical behavior. For example, oscillator has periods, amplitude, and also you have the frequency of the oscillation. So simply you specify how your genetic circuit should work. And then this is in a high level. In automated tools, you specify it in a high level, but if without any automated tool, you can just for your design workflow, you can specify this behavior, and then you search for the molecular mechanism that implement this behavior. So this is basically the circuit topology, the molecular mechanism, the interactions that could implement this behavior. I need, for example, let's say I need the production of a specific protein at specific times. So I need this kind of function. It could be performed by a genetic oscillator. It can generate such oscillation in gene expression at specific times, and you can tune the expression. I will tell you about the tuning, the expression of the oscillator, so that it can generate this protein at specific times. And also you can tune the amplitude. So you basically, you search for a topology, for a structure of interaction, a molecular mechanism that implement the functional behavior that you're looking for. We have, for example, negative feedback loop can generate oscillator behavior. This is an oscillator design. You have also positive feedback loops, can amplify the signal. Then after you find the topology, which is the abstract structure of your genetic circuit, you start thinking about what genetic sequences are optimal for this architecture. You have many sequences to choose from, but what are the sequences that are optimal for our design, that will generate the proper amplitude, for example, for the or proper period for our genetic circuit? So we think about, we need to figure out what sequences, what promoter strengths, what ribosome binding site strengths, and also terminator efficiency, things like that. You need to figure out how to do this, and to find, this is finding the functional parameter space. You have a model, I told you about modeling. You model your genetic circuit, and you start and you simulate this genetic circuit, and you analyze its behavior. And based on this behavior, based on your simulation analysis, you can make prediction on the optimal genetic sequences that will populate, or optimal genetic paths that will populate your circuit architecture. So once you make this prediction using modeling and analysis, you can then search for genetic paths that meet your specification, that meet your desired properties, like, for example, strong promoters or weak RBS. You have in genetic design, you have a library of promoters, a library of ribosome binding sites. This is ideally, but this is still under development for different organisms. But this is ideally, you have a library of quantity of genetic paths, well-characterized genetic paths that you can choose from, and so that you can model, you can tune your circuit to your predicted, to your desired behavior. And once you arrive, so we start with the specification, and we specify the topology, the molecular mechanisms that will implement our desired behavior, and then you search the functional, search for, you do modeling and analysis to figure out the functional parameter space, the range of parameters that will affect the behavior of the circuit, and you find the genetic paths for your function parameter space that populate the genetic circuit topology. And then you, once you arrive at the sequence, you can then think about optimizing your genetic sequence for synthesis, because there are some sequences that cannot be synthesized, like repeated sequences. So you need to optimize your sequence for synthesis, and to make sure that they can be synthesized. So I need to make sure that you grasp this part, because this is the main part that I will. So anyone has any question about this part?
Yes, I have a question.
Go ahead.
I have a question regarding some earlier slides. I'm sorry I couldn't speak earlier. Regarding Cello, I'm aware of this large Cello paper Chris Voigt's lab has put out. Do you know if this has been used then further by other labs, or is widely used? Because I'm not aware of anything.
Widely used. I'm not sure whether it is widely used, but what I can what I can tell you is that this tool is so robust that it make accurate prediction of the digital circuits and that match experimental data. But I'm not sure if it is widely used. Maybe I could judge by research paper. I'm not sure about the widespread of this tool, but this tool is highly accurate in its prediction.
Actually, if I can maybe
Yes.
chime in here. So, hi, my name is Prashant. So I'm actually one of the co-authors on the Cello paper, so I could probably give some context here. I think as Hatem mentioned, one of the reasons why the paper or the tool became so popular was because of the accuracy of the predictions. So it was basically a great proof of concept to show how you can accurately build genetic circuits and have a CAD tool to enable that. So that's really what made the tool popular. Now, when it comes to actually using it, so in my experience, I have had a lot of folks who have contacted me asking, because they want to use the tool to build genetic circuits, but from a computational point of view. Now, whether it's being actually used in a wet lab, I think that is slightly different, a bit more complex, because it's basically limited to the limitation of the constraint is that you do need a library of well-characterized repressors to be able to do that. Now, that being said, the Cello tool has been used in many other projects as well. I think the SD2 project, for instance, used that. It's been extended a couple of times. There's a paper that spoke about how you can build sequential logic circuits. I think that was that came out a few years back, and more recently there's one for Cello for yeast, genetic circuits in yeast as well. So I would say that I think more than actually using it biologically, I think it's basically kind of like this proof point, like an important proof point to show that it is actually possible to create a CAD tool that can accurately design and build genetic circuits. So I hope that helps.
Okay. Can I just ask one more question?
Yes,
but ideally, you would want to take the one step further and then actually use it in the wet lab to build genetic circuits. So do you think like this library part is where we currently need to build better libraries and larger libraries to then be able to actually use it for more things?
I'm not sure if I get your question correctly. Could you please repeat it again? Yes.
So I think the question that Lars asked was, is the limiting factor basically the available parts, like the constituent parts of what go into a genetic circuit? So I'm happy to give some thoughts, but Hatem, if you want to maybe take this.
The limiting factor for using this tool is the availability of the parts.
Basically, I guess, yeah. So essentially, the idea is, I mean, maybe I can just give like a few quick thoughts here. So,
okay.
In some ways, yes, I think building a parts library, like an accurate library of repressors, is absolutely important. I think mostly because it's the accuracy of the response functions of your final genetic circuit really depends on how well the repressors have been characterized. So I think that plays a big role here. But actually, before I continue, Chris also has some thoughts. And maybe, Chris, if you'd like to just chime in.
Probably along the same lines of what you were getting at, Prashant, is that I think that, and it also kind of hooks back to the earlier question about usage outside of the Voigt group. The issue really, I think, in my opinion, is that the library development is really kind of secret sauce to Voigt group. I mean, not secret in that they're really hiding anything. It's just that it's not a standardized thing that the communities agree. This is the way you characterize things, and that there's lots of libraries being developed characterized exactly the way the Voigt group has done it, which limits the applicability of the cello tool outside of the Voigt group, because you do need to have well-characterized libraries with a very specific mechanism. And one of my students has actually been struggling with this because he's trying to create parameters for modeling, because we have a model generator that we use to create models from the cello parameters. And different experiments from different people outside the Voigt group, we've struggled to actually convert them into appropriate UCF files that we can then use for our model generation. And so I think that's really a barrier here, is the fact that there isn't sort of a community agreed on standard for part characterization. The data sheets that we were promised when synthetic biology started haven't materialized exactly the way we'd like to see them. And so I think that that's an area that really needs some community engagement and discussion. And I think that would ultimately lead to a wider application of tools like cello.
Great. So thank you, Chris, for this input. So can I go ahead, or do you have any questions? Yes, any questions?
I think there are just a few more thoughts
Yes.
about this in the chat. I mean, it's really up to you, Hatem. If you want to maybe if you want to continue discussing this, that's also fine. Or if you want to continue, depending on the amount of material you still want to cover. So it's up to you.
Okay, no problem. We could have any discussion on. We could continue discussion if you would like, if you have any question. Yes. Okay, let me continue. So this is basic generic circuit design workflow. I already discussed this part. So let's discuss the key factors that control the behavior of generic circuits. I told you in the generic circuit design workflow, we start with specification and we choose the topology and we choose the parts that populate this topology. So basically, the factors that control this behavior, the behavior of generic circuits, first of all, the circuit topology, the structure of interactions in the circuit. We could have the same function that implemented by different topology, different interactions. There are many generic oscillators that have been published in literature with different circuit topology. Each one could be different in for example, in robustness, also in its in the dynamic behavior of the oscillator itself. So, and how tunable is the oscillator. So you could have different behavior of generic circuits based on the topology, which is basically the molecular mechanism implemented in order to implement in the cell in order to create the type of function that you're looking for. So we could have, for example, in the cell usually use negative feedback loops to generate oscillation, and also positive feedback loops are also available in regulatory networks. So we have the first parameter, the first factor affecting the behavior of circuits is the circuit topology. The second factor is kinetic parameters, which is basically the sequence. The promoter strengths or the ribosome binding site strengths or the ribosome binding sites are encoded by the sequence of the promoter. So the sequence of the promoter, sequence of the ribosome binding site. So basically this defines the affinity of the or between the transcription factor and the promoter, and also the affinity of the RNA polymerase to the promoter. So changing the sequence, mutating the sequence will affect the kinetics of the reaction and affect the circuit behavior overall. So we have the kinetic parameters, a factor, a very important factor of the circuit behavior. And we have also stochastic fluctuation in genetic circuits. As I mentioned, genetic circuits are regulatory interactions. These regulatory interactions happen in cells using a few number of molecules. These few number of molecules are governed by probability and probabilistic interactions. So we could have, for example, RNA polymerase and activated transcription factors and RNA polymerase bound to our promoter of interest in one cell, but in other cells, this event is not synchronized in the other cells in the same population. So you could have some genes initiated in some cells in the population, and other cells not initiating the same gene at the same time. So this lack of synchronization is caused by stochastic fluctuation. So this cell-to-cell variability is not due to genetics, is not due to is due to the probabilistic interaction, the fact that we have only a few copies of our regulators inside the cell. If we have large copies, in this case, these factors these fluctuations could be factored out. But in our case, since we have only a small copy of small number of regulators, the interactions in genetic circuits are stochastic, and hence this affects the circuit behavior. And we have also another factor, the context effects. Genetic context, basically the flanking, if we talk about the genetic context, which is the flanking sequences. That, for example, you could have a promoter that the sequence of which could interact with flanking sequences, and this affects the axis, the axis of transcription factors, or the axis of ribosome binding sites. And also you have host effects. The growth rate of the metabolic state of the host, the growth rate of the host, the biochemical environment of the host itself, and the environmental conditions that the host exists in. These are context effects that affect the circuit behavior itself, because they all come down to the concentration of the steady state concentration. They affect the steady state concentration and the dynamical behavior of circuit components. So here are some of the factors, important factors that affect circuit behavior. Anyone has any question so far? So let me carry on. So how to compose genetic circuits? We have different classes of genetic gene regulators that we can compose genetic circuits from. For example, we could control the transcription using, as we know, transcription factors, DNA binding proteins, which can be toxic, by the way, if used in combination. And also there are challenges regarding the transcription factors, use of transcription factors in genetic circuits. We have also another class of regulators that make use of catalytically inactive Cas9. This Cas9, as you know, Cas9 is used to cut DNA specific sequences, but in this case we are using CRISPR Cas9 as a targeting molecular tool. A molecule, the catalytically inactive Cas9 can simply target an RNA polymerase recruiting domain and activation domain to a specific sequences of interest using a small guide RNA. So we can use catalytically inactive Cas9 as a transcription activator, and it can also be used as a transcription repressor if it used alone without any activation domain. And this is the advantage of CRISPR Cas9 regulators, is that you can generate, because this is simply base pair interaction, RNA DNA interaction. You can simply design different interactions and generate different, and hence you could have a library of transcription activators and repressors that are orthogonal. Because one of the failure modes that I will talk about later in the presentation, one of the failure modes of genetic circuits is that you could have crosstalk between different circuit regulators. So having orthogonal regulators will allow scaling up genetic circuit design, because you have many regulators to choose from to build your circuit. So we have CRISPR regulators, and we have another class of gene regulators called recombinases. Specifically, we are interested in serine integrases. Serine integrases can catalyze unidirectional inversion of DNA sequences. The changes in the sequence can affect the RNA polymerase flux and DNA. For example, you could have the promoter flipped, and in this case, the RNA polymerase cannot initiate the downstream gene. Or you could have also the same flipping with terminator or with the coding sequence. So you simply make changes in the DNA sequence. These changes affect the transcription rate of the gene of interest in the circuit. And recombinases are known for building logic gates and also for building memory circuits. And actually, recombinases are more efficient in building logic gates even more than transcription factors, because they can generate switch-like behavior. In using transcription factors, you could have graded switching transitions. However, you need in logic gates, you need switch-like behavior. You need on-off clear on-off states. So recombinases logic gates can exhibit truly digital behavior. So this is an advantage to using recombinases. So you know about different properties of each regulator, and you choose the regulators that suit your circuit design purpose. Also, we have another class of regulator called RNA regulators. This is an adapter. This is an engineered form of regulator, just like CRISPR regulators. This simply uses a system called RNA in, RNA out. Simply a short non-coding RNA. It's called RNA out. This non-coding RNA can repress, acts as a transcription repressor of our mRNA of interest, which we call RNA in, using a row-mediated transcription termination. This mechanism, transcription termination, was a mechanism of using RNA to repress gene expression, was adapted from the tryptophanase operon. So we have our short non-coding RNA. Again, we can make orthogonal regulators from simple RNA-RNA interaction. So this is an advantage to RNA regulators, RNA in, RNA out, just like CRISPR catalytically inactive Cas9 regulators. So we have transcription factors, we have CRISPR regulators, we have recombinases, we have RNA in, RNA out. These different classes of gene regulators can be used for different purposes in our genetic circuit composition. So, any questions so far? Okay, let me continue. So, how to tune gene expression. In designing generic circuits, we need to choose the optimal generic path that generate the desired behavior. We have functional parameter space that can generate our desired behavior. This functional parameter space, we can select generic paths that we need to figure out what generic paths that populate our genetic circuit design. In our case, we use something called expression tuning knobs. These expression tuning knobs allow the control of gene expression allow the customized control of gene expression. You could change, for example, the promoter strengths. You could change ribosome binding site strengths. You could change the copy number. You could change protease tags. You could change the cooperativity of the binding of the repressor. You could have some sequestering molecules that could titrate away some circuit regulators. So you have different options. You have different options to change the expression levels of your circuit regulators. Changing the expression, in this case, you are tuning the expression level and tuning your expression level of circuit regulators to control this overall circuit behavior. There are two examples for such tuning. You could, for example, you could change promoter strengths. For example, in our case, you have a NOT gate. You need to change for a NOT gate, a NOT logic gate. You have different parameters or characteristics that characterize a NOT gate. A NOT gate simply is a genetic logic gate that gives you that if you have an on state, it gives you an off state. If you have an off state, it gives you an on state. Either way, to describe this NOT gate behavior, you have a function called response function. This response function is simply models the input as a function. Simply it gives you the output of the circuit on or off as a function of the input. So we have the shape of this response function is controlled by or is described by dynamic range of the response function defined by the on-off state and the induction threshold. The dynamic range, by the way, is the range of output as a function of input. The range of output that you can get from your NOT gate. In our case, we have on and off state. And for the induction threshold, simply the amount of the input required to reach half maximal output. So this defines the state, the on and off state. If you have, for example, if you exceed the threshold, you go to another state of the NOT gate. For example, if you have your input, you could have, and this input exceeds the threshold for the activation of the NOT gate, you could trigger the other state of the gate. And you have the cooperativity of the switch, which is simply the steepness of the curve, the switch-like behavior of the response function. You can tune the response function, the different characteristics of the response function, which is the dynamic range of the induction threshold of the cooperativity switch, based on using different expression tuning nodes that I discussed: the promoter strengths, the ribosome binding strengths, the copy number, the protease tags, the cooperativity, and the expression, also the expression of sequestering molecule. This is an example of using different expression tuning knobs to tune the shape of the response function. So for example, to change, in our case, for example, to change the dynamic range of the response function of this NOT gate, you can change, for example, the promoter strengths. Increasing the promoter strength can change, or decreasing the promoter strength can affect the dynamic range. It can change the shape of the response function on up and down. You can shift it up and down. Also, you can shift the dynamic range or shift the response function up and down by changing the copy number of your regulator, and also by changing the protein degradation, the target degradation rate. Also, for changing the threshold, you can, for example, change the ribosome binding site to change the threshold. In this case, this is very useful because when connecting logic gates, you need to adjust the threshold, the threshold of induction, the induction threshold of each gate, so that you have gates that the signal match. Because a common failure mode in generic circuit design is that you have signal mismatch. You could, for example, have a regulator expressed at a level that is not enough to induce trigger the expression of the subsequent regulator. So you need, in general logic gate design, you need to adjust the threshold of the logic gate so that the output could span this induction, the output of an upstream genetic logic gate could span the such induction threshold and initiate the subsequent logic gate. So we could change this threshold by changing the ribosome binding site. You can change this threshold by using sequestering RNA molecules. These sequestering RNA molecules simply titrate away the circuit regulator from its target site. In this case, you are affecting the concentration. You are decreasing the concentration available of this regulator, and hence affecting the circuit, the overall circuit behavior. And also another way is to use decoy operators, which simply titrate away repressors from the circuit and affect all the circuit behavior. So you can do such kind of tuning using different expression tuning knobs. You have promoter inside. This is, by the way, in transcription generic circuits, but promoter strengths, ribosome binding strengths, ribosome binding site strengths, and copy number, Buttius tags, and you have for the behavior of the circuit itself. So you change the parameters of the circuit in order to change the overall behavior of this circuit. You have another case about tuning generic circuits using a genetic oscillator. You have, for example, for genetic oscillator, you have three characteristics of genetic oscillator. You have the period of genetic oscillator, the oscillation, you have the amplitude, and the shape of the oscillation itself. So how to tune these parameters? Because it might be the case that you need a genetic oscillator that with a specific period to produce a specific protein, for example, every, let's say, 6 hours, or to produce it at specific levels. So you need to tune these parameters in order to generate a functional circuit of desired behavior. So we have different expression tuning knobs to tune the parameter of our circuit. We have the promoter strengths. This promoter strengths, for example, changes the promoter strengths as in the graph, can increase the amplitude, increase or decrease the amplitude. And also, by changing the ribosome binding sites, you can increase or decrease the amplitude of the oscillation. Also, you can change the period of the oscillation. You can change the shape of the oscillation by changing other expression, adjusting other expression tuning knobs. So, any questions so far?
I have one.
Yes.
I will read. There is a way to calculate metabolic burden or toxicity of transcriptional units or produced components, RNA, protein, or chemical, in order to inform the design to distribute the circuit on different cells, for example?
There is a
like if,
yes.
Yeah, like if my circuit is really big, there is a way to know the burden of the circuit on the chassis or host organism.
Yes, to know about the toxicity, to predict the toxicity, you mean to predict the toxicity of the regulators that you're using?
Yes. For example, when we use DCAS9,
Okay.
when you express that, it is usually toxic to the cell. So
Yes.
I wonder if there is a way to know or to inform the designer or synthetic biologist to distribute the system if needed, or to don't express that in a high gene dosage or protein dosage.
Yes, this is one way. Yes, this is one way to control, to express a toxic. If you have a toxic component in your circuit, you can express it at low level. This is one way, but you also need to tune your circuit behavior. So this is, for example, for the CRISPR-Cas9 case, I think there are strategies to control its toxicity, because you could have, for example, Cas9 alone that binds to bind non-specifically to sequences, to other sequences. In this case, you need, for example, to control the concentration of the Cas9 in the absence of the small guide RNA. So this is maybe this is one strategy that you could control the toxicity of Cas9. And also, you will figure it might not be the case that you can predict the toxicity from the beginning, but this will be subject to your you engineer the circuit and you test the circuit in the host and measure the behavior, and you could encounter different failure modes. These failure modes, one of the failure modes that I will talk about is the toxicity on the host. You can, by debugging, by measuring gene expression, you can figure out what is causing your circuit to fail. So toxicity is one of the cases. You will figure out about the toxicity of your component. If it is not well documented, you will figure it out by experimenting, by actually building your circuit, and by testing your circuit inside the host. But since we already have many engineered circuits, there are already researchers that discovered such cases and engineered the strategies to deal with these cases. One of the cases is to control the exhibition of the Cas9 in the absence of a small guide RNA, to control its non-specificity. Is this an answer to your question?
Yes, thanks. So this is more or less part of the debugging process of a circuit.
Yes. Do you know about other strategies?
No, just to, for example, distribute the system in different cells. One, yes. Another, like reduce the dosage on toxic compounds, for example.
Yes. Yes, great. Any other questions? Okay, let me go ahead. So, simply tuning the expression, the circuit dynamics. We know about the parameters that we could control, and we change these parameters to change the overall behavior of our circuit. This is, by the way, this is after, as in this case, after you generate the circuit model, after you generate the circuit, the ODE model, and you characterize your and you simplify your model, and you do simulation analysis, and using a simulation tool. So in this case, you have a working model. Once you have a working model, you can then make predictions using this model to predict the effect of different tuning knobs, different parameters in your circuits. These parameters control either the rate of production of your target proteins, of your target regulators, or the rate of their degradation. The promoter sense controls the rate of transcription. Ribosome binding site controls rate of translation, copy number, also the controls the overall census, and the degradation rate controlled by protease tags. The cooperativity controls also the expression of your genes, and also the availability of your the exhibition of sequestering molecules. So you have different tools to tune your circuit behavior, and you have the circuit behavior itself according to the circuit that you are engineering. If you are speaking about, for example, genetic logic gates, you have these different characteristics for the logic gate: the dynamic range, which is the range of the output that you can get from this gate, from this genetic circuit. This is defined by the on-off state. For example, if you have if you lower the off state, you could have a larger dynamic range. If you increase on a state, you have you can just tune this based on your different exhibition tuning knobs. And the induction threshold, I told you, this defines the input the input concentration that defines the input concentration that defines the transition between the two states before the threshold or after the threshold. Before the threshold, in this example, as I explained in the graph, you have the on state. If the input increased a little bit more, you have the off state. So this induction threshold, you can shift this threshold by, for example, as explained in the graph, by controlling the RBS strings. You can mutate the RBS sequence, or you can, there are already tools available for designing, for the rational design of RBS sequences, like the RBS calculator. You could design a small RNA that change the concentration of your repressor in the NOT gate. So you could increase the number of the repressor binding site on your promoter, or change the position. This will affect also the threshold. So you change this circuit level behavior by changing component level parameters. And the cooperativity of the switch, which is again this defines the switch-like behavior of the gate. Increasing the cooperativity makes your genetic logic gate more like digital gate. It gives a digital-like behavior. Lowering the cooperativity can give you graded analog-like behavior. So by changing the cooperativity, which again, by changing this can be affected, this can be done by changing the number, position of the operator available in the regulated promoter. So we have circuit-like behavior, circuit level behavior, and we have component parameters, component level parameters that we tune the circuit behavior. And also their example with the genetic oscillator, I already spoke about this part. So now we arrive at the why the common failure modes in genetic circuit design. Why genetic circuit designs fail? There are several reasons for this failure. First of all, the genetic circuits are simply very sensitive to their context. These genetic circuits, you have, for example, if you have your promoter characterized in a specific genetic context, specific flanking sequence, if you transfer it to another genetic context, with different flanking sequences, you could have different input-output behavior. So this kind of change in the input behavior of the promoter could affect the circuit overall behavior of the circuit by changing the expression of the component regulators. So this is the context dependency effect of the related to genetic circuit design, and also you have the context effects related to the host itself, and the context effects related to the environmental conditions. So you have your genetic circuit placed in different contexts, it can generate different behavior. So you need to characterize this behavior in different contexts. Or another way to do this is to insulate your genetic bars from the genetic context by using genetic insulators. So you simply try to standardize the sequences flanking your genetic bars. By standardizing your sequence, you get robust behavior that is regardless of the context. You have the same behavior of your or the same input-output behavior, or for example of your promoter. So you're simply looking for standardizing or insulating your genetic bars from its context. And also there is another mode of failure, is that you could have, for example, regulators expressed at the wrong ratio. You need to balance the regulator expression to generate the proper circuit response. Again, in our genetic logic gate design, you could have, for example, your circuit. If you imagine two genetic logic gates connected together, you could have your upstream genetic logic gate that produce the output at a level that is not enough to initiate the expression or the switching of the downstream genetic logic gate. So in order to make this connection functional, you need to adjust the threshold of the genetic logic gates so that they match in signals. They produce signals at rates that are enough to perform the required computation, and you have this functional connection. Otherwise, you could end up with a circuit that produce a regulator that is not enough that doesn't meet the threshold of the downstream circuit, and this way is not functional. Although it is present, is expressed, but it is not enough to trigger the threshold of the subsequent genetic logic gate. So you need to adjust the threshold. Maybe you could adjust. You have two ways. You could adjust the threshold of the genetic logic gate, of the downstream genetic logic gate, to match, for example, the dynamic range of the upstream genetic logic gate, or you could change the dynamic range of the upstream genetic logic gate to match the threshold of the downstream genetic logic gate. So again, to tune these behaviors, this circuit level behavior, you need to go and adjust the component level parameters. The promoter strengths, this is about the transcriptional genetic circuit. The promoter strengths are base strengths. Once you have a working model, you can experiment with this model in silico and see the effect of the changing these parameters on the circuit behavior, so that you could arrive at the desired behavior, whether a threshold or dynamic range for genetic logic gate, or whether a period or amplitude for genetic oscillator. So you need to balance the component regulators so you don't end up with the signal mismatch in the connection. And the other failure mode is the toxicity of the component regulators. If you have, for example, if you have a you could simply, the genetic circuits could be composed from regulators that are exogenous to the host. So it could have, these regulators could have undesired interaction with some key proteins in the host. So in this case, to avoid such unintended interaction, which in this case could affect the growth and the survival of the host. To avoid such kind of, you need to screen for this the toxicity of your regulators, and to use regulators that do not interact with your host, which will also affect the circuit itself, will affect the host as well as affect the circuit behavior itself. And this is called the crosstalk. The crosstalk of the circuit component regulators. Your circuit regulators could interact with the host, and affect the host and become toxic, and this is toxic to the host, or could also interact with each other. You could have, for example, a repressor that binds should bind to promoter A, but at the same time it binds to promoter X in the circuit. But you don't need such kind of interaction. So you need to make sure that the topology you designed is not violated by any kind of crosstalk in between circuit regulators. And also we have another kind of failure mode, which is homologous recombination. You could have the genetic circuit design, and it is working properly. It might be working properly inside the host, but due to evolutionary processes, due to natural selection pressure on the host, it could undergo sequence changes, like mutation. Or you could have homologous sequences inside the genetic design itself that you need to screen for before you construct your circuit. And this, in this case, you could have homologous recombination arrangement in the sequence genetic circuit, and you end up with a genetic circuits with different topology, with different wiring, with different sequences. So, and this of course affects the circuit behavior. So any kind of sequence change could affect your circuit behavior in some way. And this is a natural process, by the way. But this natural process happens over many generations. But it could be done very quickly if you have your homologous sequences in your genetic circuit. So you need to screen your circuit for any homologous sequences and avoid them. And also there is another common mode of failure in genetic circuit design is competition over shared cellular resources. For example, you could have your genetic circuits your genetic circuit use cellular machinery, native cellular machinery for the expression of its own regulators. And this cellular machinery include the synthesis machinery, RNA, polymerase, the ribosomes, also the degradation machinery, the chaperones. For example, there are proteins from the host itself that are being degraded by the cellular machinery, and there are proteins from the circuit itself that are also to be degraded. If you have some kind of queuing in the degradation process, and this not only affects the host but also affect the circuit behavior. For example, it is known in dynamical circuits like oscillator that these dynamic circuits require a rapid degradation of its regulators. Otherwise, you could end up with a dysfunctional oscillator. So in this case, if you have already your circuit uses the same machinery, the shared resource, the same machinery as the host, in this case, you could have some delay in the degradation of the circuit components. And in this case, this will in turn affect the concentration of your regulators. You need for the oscillator, it needs to degrade its repressor very quickly. However, because the degradation machine is a shared resource, you in this case you have a delay in the degradation and an accumulation and unintended, undesired accumulation of your repressor, which in this case will affect the circuit behavior. So in this case, you need to, for example, there are some strategies that use orthogonal machinery to the host. This one way that could engineer robustness into circuit and genetic circuit design. This also, there are another way of failure based on this kind of shared resources. For example, this is in the circuit itself, and this is called retroactivity. What happens in this in this kind of failure is that you have, for example, as two downstream circuits, two genetic circuits connected together. The downstream circuit is connected by some regulator from an upstream circuit. This regulator is titrated away from by binding and initiating the exhibition in the downstream circuit. This regulator is in effect titrated away from the upstream circuit, and in this case it affects the dynamics of the genetic circuit. And if you have of the upstream genetic circuit, which might be, for example, an oscillator that synchronize the operation of different downstream genetic circuits. If you connect more downstream genetic circuit to this oscillator, you have the repressor titrated away from this oscillator. In this case, it could it will affect the oscillation itself. So you don't have insulation in behavior in this case. This is this kind of context effects at the circuit level. You connect more downstream genetic circuits, and these downstream genetic circuits titrate more regulators from the upstream circuit, which in my example is an oscillator, and by titrating away, by binding this repressor binds to the downstream circuit, more downstream circuits, and in this case it is titrated away from the upstream circuit, and hence affect its concentration available for the oscillator to function, and hence affect the oscillation itself. So there is an engineering strategy for dealing with this kind of retroactivity, which is by a separation of time scale of the reaction. I'm not totally familiar with this kind of strategy, but simply they insulate the effect of the downstream genetic circuits on the upstream genetic circuits. So you have some kind of insulation between the upstream and downstream genetic circuits, so that you have oscillation, and at the same time you have the downstream genetic circuits controlled by this genetic oscillator. So this kind of insulation will help you engineer more robust circuit behavior. So I think this is overall of the my presentation. So any question?
Less so a question and more so just interested in discussing, given the excessive number of ways you can optimize the circuit and the excessive amount of ways that the circuit can fail, sort of, is anyone aware of where we stand at our ability to create implementations that are actually control useful biological functions, and sort of the timescales that it takes to, you know, perform and design an implementation?
Yes. Could you please repeat your question again?
Yeah, so just like examples of successful genetic circuits.
Yes.
That more so than like a toy design or a proof of concept, actual examples of genetic circuits that can perform useful functions.
Yes.
And if anyone's aware of like how long the design build tests, you know, cycle in total takes, right? Years, you know, 3 years, 4 years, you know, 12 months. I'm just curious as to how good we are at this, because it seems
how good we are. Okay.
Yeah, or there's so much here, right, that you can do.
Okay. Actually, there are more complex genetic circuit designs that have already published in the literature, more than the examples that I give you in the presentation. These are only the genetic logic gates and oscillators are simple examples of the genetic circuit design that we have accomplished. There are more sophisticated designs that are already published in literature that, for example, control the pattern formation in differentiation and in tissue engineering. You could have, for example, a living cell that interprets a morphogen gradient in the environment, and based on this stem cell, and based on this morphogen gradient, it can, based on the concentration of the morphogen in the environment, it can initiate differentiation. It can make a decision regarding its fate. With high concentration of this morphogen, you could have a differentiation, a certain fate decision. With another, with less concentration of the morphogen, you could have you could have another fate decisions. So these pattern forming circuits are already implemented in CRISPR, using CRISPR regulators, and also it has been implemented using transcriptional regulators, using a kind of motif called incoherent feed-forward motif. This incoherent feed-forward motif can generate different behaviors based on the morphogen concentration in the environment. And this is one example. Another example is also at the level of tissue engineering, more sophisticated example, using building artificial tissue anesthesis system, which is basically you have stem cells and you have beta pancreatic cells. You program stem cells to generate more of the beta cells in case this beta population are attacked by the immune system. In type 1 diabetes, they are attacked by the immune system and they're depleted from the body. So you need to generate more of this beta pancreatic cells. At the same time, you need to maintain your stem cell population. So you need in this circuit, they coupled different genetic circuits that are already designed, like toggle switch, like a pulse generator. They connected these foundational genetic circuits to build a more complex behavior. So in answer to your question, our ability to design genetic circuits is improving with time. We don't have we don't match, our design capability doesn't match the capability of natural genetic circuits, but it is already improving with the availability of more computational tools, more libraries of regulators, orthogonal regulators, and also more abstraction in our design. As I told you in the beginning, that we still we have enormous potential of synthetic genetic circuits that are still unharnessed, but we need to develop tools, and these tools that can automate and abstract the circuit design. And also, more importantly, is to learn how to design circuits at a larger scale. This is called the design principles. We learn the design principles of genetic circuit design by building circuits and failing and understanding why the circuit fail, and try to implement using this by learning from these failure modes, we could design more robust circuits, more circuits that are tuned to our desired behavior. So again, our ability to design is improving with time, but we have already accomplished more sophisticated versions, like the examples I give you, that reflect our improvement in genetic circuit design.
Fantastic answer. Thank you.
You're welcome. So, any questions? There's a way to calculate metabolic burden to sustain. Actually, there are. I'd like to let you know that this is not what, because of time restriction, I didn't go over some points, like explaining how to model and simulate and analyze genetic circuits, how to go from, for example, in this case, how to go from circuit topology. First of all, how to select a circuit topology, because this is very important. You have a function, you have a desired function, and you need to implement this function. You need to figure out what kind of interaction networks that your genetic circuit need to perform in order to generate this function. Maybe this is pretty obvious in designing genetic logic, in designing digital circuits, because it is all very obvious that you need that, for example, if you need a specific computational operation, you can this computational operation is defined by Boolean logic. This Boolean logic can be implemented by genetic logic gates, and you can have this workflow automated using a GDA tool like Cello. No wonder why it is no surprise that the first genetic design automation tool was built for digital circuits, because for digital circuits it's amenability to design, it's designability, and it's also amenable to automation. So you could have such kind of automation build not very easily, but at least much better than other kind of circuits. For example, it is more challenging to design an automation tool for dynamical circuits, like an oscillator, for example. Not only oscillator, but also more sophisticated version of the dynamical circuits. This is dynamical behavior. You have in digital circuits, you have simple logic. You could design complex logic, but you have on off. However, in dynamical circuits, you have, for example, dynamical characteristics like the amplitude. You have the oscillation of the circuit. This kind of dynamics is not pretty straightforward to automate using computational tools, and also analog computations as well is not amenable to, you can, although analog circuits, as you know from electronic engineering, analog genetic circuits can make computations very more efficient, more than digital logic circuits. However, these analog circuits, the ability to design analog circuit and to automate the design of analog circuits is not straightforward as digital as automating digital circuits. So we have challenges with the automation of different kinds of genetic circuits. So our ability, our engineering strategies, as our ability to engineer circuits and also to learn about the different design principles, we could that describes the operation of different genetic circuits. As we learn about the design principles of genetic circuits, we could simply set these design rules in a computational design tool, and these design rules then can be used to design our difficult to automate genetic circuits. For example, it is known for genetic oscillator that you need for robust genetic oscillation, you need, for example, high cooperativity, or you need strong promoters. So this kind of predicted design rules. In this case, you could implement these design rules in computational tools to help you automate the design. But also, as part of the automation, you need to abstract your design process. You need to build the foundational genetic circuits and have your computational tool to wire these foundational circuits to reach the desired behavior. In our case, in the case of cello, they have already built a library of genetic logic gates. These genetic logic gates were available to the cello in order to be wired together. Once you have the logic table or the twist table available, you can draw on your library of genetic logic gates to build your complex digital genetic circuit. So you have a library of well-characterized genetic logic gates that is used by the automation tool to build your more complex digital circuit. So we need to implement the same concept in other kinds of circuits, but this needs to be this will improve with time, as this kind of automation is not very straightforward as in digital genetic circuit design. So, any questions?
Actually, not
Yes.
more of a question, but more of I just wanted to take a minute to actually to give a shout out to this workshop. Hatem, I think this is really great. As someone who's been doing genetic circuit design, it's really amazing to see one compendium of all these topics, all these concepts in one place. So I just wanted to take a minute and say this is a great job. This is a great workshop. So thank you so much for putting this together.
Yes, you're welcome.